so I moved recently last year from the Curie Institute in London to the University of Geneva. And so I'm a theoretical physicist interested in tissue morphogenesis. And so I'm going to tell you about physics and its role in the morphogenesis of tissues. And I can already say, please stop me anytime to ask questions during the talk. Okay, and so let me start by a very broad introduction, which is that something that physicists are very familiar with, which is that in a physical system, one can consider, describe a physical situation at different scales. And one can think about a given physical system at the microscopic scale and think about the molecules, the interacting molecules. And usually there are many of them which interact together. Or one can choose instead to move to the macroscopic scale, for instance, defining these boxes here, and then inside each box there are many of the microscopic molecules. But on the large scale, we have fields like a concentration or a pressure, which define what is inside these boxes. And these fields, they vary on very large spatial scales. And usually, the equations that, or the physical behavior that describe what's going on at the macroscopic scale is, in fact, simpler and independent of many of the microscopic details of the interacting molecules. So many of the details of the macroscopic system become irrelevant when we want to describe it at the macroscopic system, at the macroscopic scale. Okay, for instance, if one wants to describe a gas, okay, one does not describe every molecule inside the gas. Instead, one may want to choose a macroscopic approach. Okay, and an example of this, which is also familiar to physicists, is the hydrodynamics of simple fluids. And this is also a simple problem that you can solve analytically, which is the question of the flow of a fluid inside a pipe. And so you can consider a cylindrical pipe. There is an applied pressure. And then you can write down the equations of hydrodynamics, the Navier-Stokes equation, and find out that there's going to be a quadratic flow profile with a maximum velocity in the center. And you can calculate the magnitude of this velocity. And it depends on some geometrical parameter that describe the tube. It depends on the applied pressure. And in fact, there's only one parameter that really describes the fluid, which is the viscosity. And so in the end, it doesn't really matter how complex the fluid is, and if it's made of water or honey, eventually, if it behaves as a Newtonian fluid, the final outcome is that everything can be described just knowing the viscosity, okay, one parameter. So now, following the question is, can we follow this kind of idea in a biological system? Okay. Okay, so the movie is not working. Okay. Okay, so what this movie is supposed to show is the development of a fly. So is this about the movie or no?
No, it's about the mic.
Okay.
Sorry.
So what this movie is supposed to show is the development of the pupal wing. And okay, it's a pity because it's really a beautiful movie. So this is the wing in the Drosophila at the pupal stage. And so it's this big tissue here, which is made of thousands of cells. And when you, at the end of so initially the tissue is rectangular, the movie is unpatterned. And then as time progresses, there is the establishment of a pattern on the wing. Yeah. Great, thank you. Okay. Okay, still doesn't seem to work. Okay, but the movie doesn't play. But okay, I don't know if something can be done or no. Okay. Okay, and what we would see in this movie is that all these cells are collectively moving, and as they collectively move, they deform the tissue, and which in the end takes the shape of a wing. Okay, and what's also so here, what you can see in this movie also is that this wing is made of many cells. There are thousands of cells. And if you look at the large-scale flow, you don't really see in each individual cells. It looks like a macroscopic object. And there is a similar question of how much one can say about moving from the cellular scale to the macroscopic tissue scale. So if we compare this to a physical system where, like a gas, where there are 10 to the 23 particles, of course we are not at this order of magnitude. There are thousands of cells, not millions or millions of millions of cells. But still, one can ask if one can describe this system on the scale of many cells. Okay, and if one thinks about this question, there are several things to take into account. One is mechanics, and the fact that because the cells are moving, one needs to describe the mechanics of the system and the fact that cells are exerting forces on each other. The other thing to consider is signaling. So cells can secrete molecules, which then diffuse in the extracellular space, and then the molecules bind to receptor, and then the cells can undergo information processing and use the information that they get by measuring the extracellular concentration of molecules to take decisions and form patterns. So if we want to understand tissue morphogenesis, we have to deal with these two aspects, mechanics and signaling. Okay, and here I'm going to talk mostly about mechanics, and at the end I'll talk a little bit about signaling. Okay, if we want to think about mechanics, I would argue that the right framework is to use active matter physics, which is a branch of physics which has developed massively in the last 20 years. And here, okay, I'm defining it as the physics of systems which are not living, so which are dead, but which can be brought out of equilibrium because they have a source of energy at the macroscopic level. For instance, a set of particles which are on a substrate and then can self-propel because they can use a chemical reaction to do this. And already from this definition, it's clear that there is a close relation between active matter physics and biology. One of them is that usually in this active matter physics system, one is interested in many components, and similarly in the cells there are many molecules, or in the tissue there are many cells. If one wants to create an active matter system, one needs to find a way to inject energy in the system. So one way, which is described in this paper, is to take these particles here, which are coated, and then use laser irradiation to heat them up, which creates a gradient of temperature, which makes them self-propel. And so here the energy comes from the laser that's injected into the system. In a living system, instead, of course, there are neutrons that the cells can use. And inside the cell, the source of energy is the hydrolysis of ATP, so the cells use nutrients to make ATP, and this reaction here from ATP to ADP is releasing energy, and this can be used by the cell to drive processes out of equilibrium. Okay, another important aspect is that one does not necessarily deal with systems of particles which are isotropic. They can be that the particles are anisotropic, in which case one can have something like a liquid crystal, where there is a level of order inside the system, so a broken symmetry variable. Okay, and one could create this artificially by making an active matter system made of elongated molecules. But in the cell or in the tissue, this also occurs naturally because cytoskeletal filaments are elongated, for instance, or because the cells are not necessarily isotropic. And talking about this more precisely, so if you have a nematic, that's a type of order in a system where there are particles which are elongated. But they still have an up-down symmetry. So it's like a cylinder. Okay, you have a direction, but you don't have an orientation. And this kind of order can emerge in a tissue because the cells are not spherical, they are elongated, and then one can associate an axis to each cell and ask if there is a global nematic order emerging from this. Okay, and another type of order is the system made of polar molecules. So where now the molecules have an axis, but they also have an orientation. And similarly, in the cell, there are different proteins inside the cells can distribute themselves anisotropically in the cell. And this is what happens in planar cell polarity, for instance, where you have this green molecule on one side, red on the other. And then this defines a polarity axis inside each cell. And if this polarity axis is coherently oriented in the different cells, that defines a large-scale polarity order in the system. Okay, and now I'm going to show two examples about the emergence of this order. One is actually the work from Pascal. So in this very nice, okay, the movie is not working. Okay, so in this, what we are supposed to see here, so this is a plate where one puts these elongated cells, mouse embryo fibroblasts, and then they divide, and they form a pattern where the cells are elongated. Okay, and this is a quantification of the elongation pattern. So each of these lines indicates in which direction the cells are oriented. Okay, and what? Maybe this is working somehow. No, okay. So, and what happens in the end is that the cells tend to align themselves parallel to the axis of the boundary of the disk. And this pattern here, which is still quite complicated, simplifies. And it simplifies with the emergence of two topological defects at the end of the... maybe I can try to draw it. Yes. So they're oriented like this. Okay, and here, there is a topological defect, so a point where the incoming lines are not coherently arranged, and so in this point one cannot define an orientation, because if one does a turn here and follow the rotation of the order, there is an overall rotation of 2 pi of pi, so 2 pi divided by an half. So therefore, this is called a defect 1/2. Okay, so a defect +1 is just an aster like this. But the nematic defects have this structure here where they rotate by pi instead of 2 pi. Okay, and this kind of organization, so because the cells orient parallel to the boundary of the disk, there must be a total topological charge, so a total number of defects inside the pattern, which must be +1. Okay, that's a requirement from aligning the cells parallel to the boundary of the disk. And then one can do a calculation, one can ask, okay, there is this topological requirement, now what is the field going to do? And one can write so if n is a vector, the orientation vector at a point inside the disk, one can write a distortion energy So the idea of this distortion energy is that it tends to minimize the gradient of the polarity. So that an organization like this, not of the polarity, of the nematic order, so if one has a part of the system where the nematic axes are parallel to each other, this is a low energy, in fact, zero energy configuration. And if one tries to create a spatial gradient of the order, then this becomes a high energy. Okay, this is what this expression means, is the Frank free energy. Okay. So this is something that is known from passive liquid crystal, because it's a relatively simple idea. The system doesn't have to be active. Okay, it's just there is generally a cost to create spatial distortion. And then if one applies this logic here and calculate the configuration of minimal energy, one finds that there are two defects of charge plus 1/2, which appear close to the boundary of the disk. Okay, of course, there is an invariance by rotation, so these defects can be anywhere if I rotate them like this. Okay, but all these solutions minimize this energy. And this is actually what happens with this experiment. In the end, the system creates this pattern with these 2 plus 1/2 defects. And so, is it clear? And so that's a very nice experiment because it shows that even though the cells are extremely complex objects with many things going on, many active processes, in the end, if you culture them on a circular plate, in this example, at a large scale, they can just be described by the equation for a passive liquid crystal. Another example from a recent work of Benoît Ladoux. Okay, so here, there is so here they culture these cells, MDCK cells, on an annular ring, okay, and the cells divide, and as they divide, they form these little trains of connected cells. And these cells are polarized. This is what the right image is showing. One can follow the activity of RAC1, and inside each cell, this activity has a front and a back. So one can associate a polarity axis to each cell, which denotes the direction of RAC1 activity. Okay, and it turns out that so these cells are moving, and they are moving in the direction that depends on this polarity axis inside the cell. And at the beginning, they form these little trains which can move in different directions. Okay, and as they divide at some point, these different trains, they have to connect. And once they fill the annular ring, so every time they contact, they somehow synchronize and they flip their polarity so that at the end of this division process, all the polarity axes are the same. And there is an overall spontaneously broken symmetry with a rotation of the entire train of cells on the annular ring, which chooses here arbitrarily a direction of rotation. So what that shows is there is a polarity axis in general in cells, which can be caused by many proteins, but here it's by RAC1. And there are also mechanisms that allow the cells to communicate their polarity axis and synchronize them on a larger scale. And this is what creates here this overall polarity pattern on the ring. Okay, and to show also, you can imagine what's going on here. Here they have cultured the cells on different substrate with different shapes. And no matter the shape of the substrate, because of course it's not the fact that it's a ring that matters, this train of cells synchronize, and then they all start to rotate with constant velocity on the ring. Okay, so now the first thing I would like to discuss, so these examples, they deal with tissues which are cultured on two-dimensional substrates. But in practice, in development, many times there is the question of three-dimensional deformation of tissues, which are necessary to make up the body plan of the organism. Okay, we are not flat, so we must three-dimensionally deform to get our shape. So I'm going to discuss this and introduce the framework of active surfaces, which is meant to deal with the description of how tissue deforms in 3 dimensions. Okay, but first let me start with a biological example, which is work we have done not really on morphogenesis, but on cancer biology. And so we have worked with the lab of Axel Behrens in the Creek. And what we have done here is look at the growth of pancreatic tumors in mouse, okay, because this group is interested in cancer biology, and so is using the mouse to induce tumors and study how they behave. Okay, and then they have this imaging technique that allows them to image the pancreas in 3 dimensions. So this is the 3-dimensional pancreas, and this is a zoomed-in view. And what you can see in the zoomed-in view is that there is a tree of tubes. So the pancreas is made of this tree, and as you get deeper into the tree, the tubes get smaller and smaller. At the end, they are capped by acinar cells, which secrete enzymes, which flow in the tubes. Okay, that's the structure of the pancreas. And then they have models, mouse models, meaning that the mice are genetically modified so that if one injects tamoxifen, some of the cells in the pancreas will randomly react to this, and this will trigger a genetical modification. Here there will be the deletion of an oncogene, this FBW7, and the deletion of a tumor suppressor gene, and the activation of an oncogene. So essentially, this cell becomes cancerous. Okay, and then one waits, and normally the cells of the pancreas are dividing very rarely, but these cells, being cancerous, divide much more often. And then they form after a few weeks, they have grown, and they form a small clone, which is an early tumor, which is the beginning of the growth of the cancer. Okay, and this movie here is showing that we can image in 3 dimensions what the shape taken by these tumors. Ah, okay, wonderful. Okay, because it's the best movie. So, okay. So you can see in 3 dimensions you can see the tube here, the pancreatic tube, and you can see the red color, which corresponds to the cancerous cells. Okay, so one can see where they are, and the fact that here they have formed this little bulge of cells. Okay, and then if one looks at these tumors in more detail, one can see that they have different shapes. In some cases, like is shown here, this is a 3D view, this is a cross-section. In some cases, they grow to the inside of the tube. In some other cases, so the yellow outline here indicates the boundary of the tube. In some other cases, they grow to the outside, so they locally enlarge the size of the tube. Okay, so different shapes. Sometimes they grow to the outside, sometimes they grow to the inside. And then one can ask where you can observe these deformations of the tissue. And what this plot is showing is that this is strongly correlated with the size of the tube. So if you look in a small tube, they grow to the outside. And if you look in a large tube, they tend to grow to the inside. Okay, so at different places in the pancreas, they grow differently. And so this looks like a question of mechanics. And so we can then turn to our methods to understand tissue mechanics and morphogenesis. Okay, and of course we have to think about what is important about the tissue mechanics. And what's important is that the tissue lining up this pancreas is an epithelium. Okay, so it's made of cells connected to each other by adhesive links. And inside each cell, there are filaments and motors. And these myosin molecular motors and actin filaments notably exert forces on the surface of the cells. Okay, and usually these actomyosin networks are mostly present on the cell surface. Okay, and then the forces generated by the cytoskeleton deforms the tissue. Okay, and the fact that this myosin molecular motor exert forces, it results in a surface tension. Okay, because these myosin molecular motors, they tend to constrict the actin network. So I'm showing 2 movies here or maybe not, to illustrate what happens. So, okay, so this is an image of the surface of a Drosophila embryo, and one can see in red the cell outlines, and in green this one has labeled the myosin molecular motors. And it turns out that in this movie, the myosin molecular motors are locally pulsating, and as they pulse, every time there is a pulse of myosin, one can observe a motion of the lateral cell interfaces which are connected to it. So that sort of visually reveals that these myosin molecular motors are driving a constriction. Okay, and another classic experiment which shows that there is tension in a tissue is this laser ablation experiment. So here this is again a tissue in Drosophila, and here one comes with a laser and ablate the junction, as a result of which there is a recoil of the junction, which reveals that if one thinks about the balance of forces in this tissue, there is actually a tension on different interfaces. Okay, and if one does a laser ablation, one releases the tension on the junction. Okay, so based on this, we thought about looking at the distribution of this myosin molecular motor, here the phospho-myosin light chain, in the tissue, in the tumors, and outside the tumor. Okay, so this is the cross-section, this is the tumor, this is the outside, and here this is the myosin imaged. And if one quantifies the distribution of myosin, so there are two peaks. These two peaks, they correspond to the basal and apical surface, where most of the myosin accumulates. And the peaks have different amplitudes in the wild type and in the transformed cancerous cells, which indicates that there is a redistribution of myosin. In the wild type cells, most of the myosin is apical. In the transformed cells, there is a redistribution. The myosin goes basal, and the apical level decreases. So that gives a picture of the force distribution in this pancreatic duct, where there is apical. The apical side of the tissue is facing the center of the duct. There is basal on the outside. And based on the distribution of myosin, one expects that there is a tension which is larger apically than basally. And this is modified in the cancerous cells, which divide and also redistribute their myosin so that the basal and apical levels become more equal to each other, which also suggests that the surface tension becomes more equal to each other. Okay, now the question is, what's the deformation that's going to result from this redistribution of forces? So one possible approach is to use a vertex model, and here in particular a 3D vertex model, where one represents the tissue by a set of vertices, which are at tricellular junctions. And in order to capture the three-dimensional aspect of the tissue, these vertices, they can be apical or basal. And one can then, it's a sort of minimal description of the geometry of a tissue, because just by giving the position of the vertices and which vertices enclose the which cell, we have a definition of, simple definition of the tissue geometry. Okay, so, and the vertices can move in 3 dimensions so that the tissue can deform in 3 dimensions. Okay, and then in order to have a mechanical description of this tissue, we need to we need to find a way to implement our knowledge of cell mechanics into this model. And this can be done by introducing a virtual work function, which contains several terms. One of them contains the cell pressure, which is coupled to the variation of the cell volume. And this internal pressure, here we adjust it so that the cell maintains its volume, because we are looking at examples where the cell volume is not really varying. There are also surface tension on different interfaces. So this surface tension is coupled mechanically to a change of area. And this represents the active tension generated on different interfaces. And there can also be line tension, like in this laser ablation experiment, where one cuts a junction. One can see that there is a line tension specifically at the cell junction, because there are specific structures which are not only on the cell surface, on the entire cell surface, but also on the cellular junctions. Okay, and then having formulating this, one can calculate the force acting on each vertex, which comes from all these elements: pressure, surface tension, line tension, by differentiating the virtual work with respect to the position of a vertex. Any questions so far? Yes?
Sorry. What does virtual work mean?
So it's a good question. So it's one possible way. So one possible way of expressing, so when you think about the mechanics of a problem, you can write force balance, so the law of Newton. You can also write, we can ask how the system would behave if you do an infinitesimal displacement of the points, and that then creates this virtual work. Yeah, okay.
Is there any thought put into how many vertices you choose? I mean, like, why do we choose a hexagon, or why not something more complicated?
Okay, yeah, good question. So, so in principle, when you simulate this model, it can happen that the junction, the 2 points on the junction become very close, and then there is a topological transition. So, depending on the model that you choose, whether you put more noise, how you distribute the tension, these topological transitions are going to change the topology, so the number of neighbors of each cell. So this is not something which is imposed. This is something that comes out of the choices. Okay, and so here we in order to simulate these tubes, these pancreatic tubes, we have generated cylindrical tubes with a different number of cells on the perimeter, because we went in the pancreas, we measured the number of cells on the perimeter of the tube, and we see that this number, bigger tubes simply have more cells. The size of the cell is not really varying in different regions of the organ. And then we do a simulation where we pick a cell in this tube, and we do what we think happens in the tumor, meaning that this cell is going to grow and divide and generate a small tumor. And also there is a redistribution of surface tensions, apically and basally, like we saw from the myosin measurement. Okay, and if we do this and look at the simulation result, we see the same thing as in experiment, which is that in a small tube, the tumor grows to the outside, and in a big tube, without changing anything, so with the same mechanical parameters, the tumor is growing to the inside. This is a cross-section, a sagittal view compared to an experiment. Okay, and one can actually even test the quantitative comparison, and one finds that quantitatively the deformations match without much effort. So that's one perspective on the problem, which is to describe the system at the cellular level. Okay, but then we have these simulations, we have these rules, but that doesn't give us a direct understanding of the origin of this behavior, of why. So we see it in the simulation, but that still doesn't tell us why the tumors grow to the outside or the inside. So instead, one can choose to move to a description of the system as a surface. And this is what I'm going to describe now, is a theoretical framework to describe the mechanics of polar and nematic active surfaces. So let me start by something quite general, which is the framework of linear thermodynamics close to equilibrium. So the question here is, so we know that we have thermodynamics, we can describe an equilibrium system, okay? But there is also a framework to describe what happens for a system which is not too far from equilibrium, and to obtain equations that's going to tell us how it behaves. Okay, that's the framework of linear irreversible thermodynamics. So the idea is, there is, maybe I will do it on the board, actually. So the idea is there is a thermodynamic system with a free energy. Okay, this system could be a gas, it could be a piece of living tissue, and there are a set of thermodynamic variables that describe the state of the tissue, the state of the system. For instance, this could be a concentration, this could be a volume. Okay, different thermodynamic variables that you can use to characterize the system. And okay, and then if I differentiate the free energy with respect to one of these variables, I get a thermodynamic force. This is a state variable. This is a force, a thermodynamic force. It's not necessarily a mechanical force. Okay, but okay. So for instance, if I have a concentration phi, the coupled variable f is the chemical potential. If phi is a position of an object, then the coupled F is really the mechanical force on the object. Okay, we can continue the list like this, but so in thermodynamics, there is the idea of having the state variable and the forces which are conjugated to them. Okay, and okay, so that's just a basic definition saying that the differential of the free energy can be expressed in this way. Okay, and then if the process is sufficiently slow, I can calculate the entropy production in the system. By just taking, if I do things sufficiently slowly, this will apply quasi-statically, and I can just take this equation to differentiate the. Sorry, I should have said here I use I. So I assume implicit Einstein summation. So if I do it explicitly, I have a summation here. Okay, and then if I take the time derivative, then I find that the entropy production, so the sum, so the time derivative of the free energy of the system is a sum of thermodynamic forces times fluxes. So the fluxes are just the time derivative of the state variable. Okay, and now I don't know in general how what these fluxes are, but I can postulate something. So when I write this, I postulate that the forces are linear functions of the forces. The fluxes are linear. I do an expansion of the fluxes into forces. So the idea is that if I apply a non-equilibrium force on the system, that's going to result in a flux, because if I have no force, the system is at equilibrium, and so the thermodynamic state variables are not changing. Okay, and then I introduce this matrix, which are linear matrix. I don't know what the coefficients in it are, but this is the part that is postulated. So they are called phenomenological coefficients for this reason. Okay, so this equation doesn't come from first principle. It's looking at the system already at the macroscopic scale, and then writing that in general the flux are function of the forces, and one can linearly expand. And if one expands fluxes into forces, that's generate this matrix. Okay, and one example that relates to what I was saying of the beginning of this approach is the equation for a Newtonian fluid. Okay, and in this equation I have a stress and I have a gradient of flow. And here I have a viscosity. So in this kind of structure, I have here the stress plays the role of the flux, the gradient of flow plays the role of the force, and in between one introduces the phenomenological coefficient, something which is not known a priori, which relates the two. And it sounds maybe a bit simple, but in fact it works really well because all the hydrodynamics, the description of water and air are all based on first writing down this equation, which is postulated. Okay. Is it clear? Yeah. Of course, once we have defined this coefficient, one can measure them experimentally. Okay. Okay, so now this is what I want to do, but I want to do this in a specific setting. I want to describe a surface that's going to be a model for an epithelium. And so I'm going to choose a situation where, okay, I have a surface. This surface can be curved. So I'm going to use the differential geometry, so the mathematical tools that allow to describe curved surfaces. There are two fundamental objects that I need to use to describe curved surfaces. One is the metric tensor. It's a quantity that tells us when we move on the surface how distances change. The other one is a curvature tensor. It tells us about the three-dimensional shape of the surface in space, so how much the surface is curved. Okay, and this surface is also deforming, so I can have a velocity field. The normal part of the velocity field tells us about the deformation of the surface, and the tangential part, it tells us about how the system, the deformation of the system on the surface, and these deformations, they define a gradient of flow, the spatial derivative of the velocity. Okay, there is also, the surface is active, so there's going to be a reaction rate that describes, for instance, how the surface is consuming ATP to work out of equilibrium. And because we want to describe epithelia, there are also broken symmetry variables, for instance, a nematic field or a polarity field on the surface. Okay, and now I want to use the framework of linear thermodynamics to obtain equations that's going to relate these quantities to each other, and that's going to be a model for an active surface. But before I do this, I need to think about symmetries of the surface. I need to think about what actually the surface looks like. And one can do a categorization of surfaces based on their symmetries. And we have found that if we look at isotropic, polar, and nematic surfaces, there are 19 types. Okay, so there are 5 isotropic types, 7 polar, and 7 nematic. Okay, so where does that come from? What does that mean? So, okay, if we think about an actual surface made of molecules, one can take a piece of the surface, this is this blue box, and one can ask how the piece of the surface is going to change under different symmetries. And some of these symmetries which are particularly relevant for a surface are a mirror symmetry with respect to a plane tangential to the surface or perpendicular to the surface, or a rotation by pi, which flips the up and down side on the surface. So in principle, I can take a piece of the surface, apply all this transformation, and ask how the surface is going to change. And if I do this, I find that there are 3 pseudoscalars that can be associated to a surface. Okay, what do I mean by this? So this pseudoscalar is a value, is a number, which quantifies a broken symmetry. And it's a pseudoscalar because it doesn't transform in the same way under different symmetries. One of them is this value here is the up-down, epsilon up-down. It describes the up-down broken symmetry. If it vanishes, it means that there's no difference between the up and down of the surface. And if it takes non-zero value, it says that now one can distinguish up and down. I can also define a chiral broken symmetry, which is a coefficient. Again, if it vanishes, it says that the surface doesn't have a chiral broken symmetry, and if it takes non-zero value, then there are chiral molecules inside the surface. And there is a third type, which is distinct from a chiral surface, but which I think is sometimes mixed up, which is a planar chiral surface. So here the idea is that I take a surface and I can draw a loop on it, which tells me what clockwise is and what counterclockwise is. Okay, and this is not the same as chiral, because if I take a surface and I draw this loop, and I rotate the surface and I look at it from the other side, the loop is rotating in the other direction. So because of this, it's not a real chiral object. It's a different type of broken symmetry. Okay, any question? Okay, so I'm giving here some examples of surfaces out of this. Okay, so I should say, so using this categorization, that's how one can find that there are 5 types of isotropic surfaces, and then one can do a similar analysis if there is a polar vector or a nematic tensor and find these additional types. Okay, and I'm not going to show the 19 types, but just some example. So if I have an isotropic membrane bilayer, the up and down surface are made of phospholipids. There is no broken symmetry. And if the molecules are not chiral, it's not chiral, so it's the simplest type of surface, which is simple isotropic. It's also possible to generate monolayer of lipids which are tilted. And this is actually a polar up-down broken symmetry surface, because when the molecules are tilted, that defines the polarity vector, which is tangent to the surface, so it's a polar surface, and also it has a broken up-down symmetry. And if you think now about a bilayer with tilted molecules, it's a strange type, actually. It's pseudopolar, because if I choose one layer and associate a vector to it, I can see that if I choose the other layer, I will take the opposite vector. Or say it differently. If I define the vector as going on the right based on the upper layer, if I apply a mirror symmetry with respect to a plane tangent to the surface, that flips the polarity of P. So this kind of order on the surface is not really a polar order, it's a pseudopolar order. If we think about the surface of a cell, which is made of myosin molecular motors, membrane, polar actin filaments, which are actually polar and chiral, this is the scenario where all symmetries can be potentially broken. The system can be chiral. It definitely has up-down broken symmetry. And if the filaments align, it can also become polar or nematic. Okay, and there are also phases in this list which, for which I don't know of a physical realization. One example is to have rotating polar motors. I don't know of an example of this. In biology, there is the F1, F0 ATPase, which is a motor that embeds in a membrane and rotates. But it's not exactly this phase because it's a rotating motor, but it has a broken up-down symmetry, which this phase doesn't have. Okay, and another example is a pseudonematic phase, which is made of 2 parallel layers with orthogonal nematic orientation. And it's pseudonematic because if I choose, you can see I cannot choose a single axis. If I choose an axis here, the axis on the upper layer, the axis on the lower layer is different. Actually, I will show later a possible biological realization of this phase. Oui. Yes, that's my example exactly. Okay, and then so now I have described my different phases, and now I consider a surface, but I want to describe an epithelium. So I'm not going to consider that the surface has an infinitely small thickness. I'm considering that I'm describing a system with a finite thickness by a simple surface. What this means is that if I think about the three-dimensional object and I do a cut of the surface, there is a distribution of stress. If I sum these stresses, that give rise to a tension. But the stresses are not necessarily the same along the cross-section, and therefore there can also be, in general, a torque that acts on the boundary, on the cross-section of the surface. So it's a force that tends to make the cross-section rotate. Okay, and if once I have defined these tensions and torques, I have a force balance equation, I have a torque balance equation, and here I'm at low Reynolds number, so this is the equivalent of the law of Newton with the sum of forces equal to the inertial term, but inertia is negligible in general in this, at the scale of the system I'm showing here. Okay, so then I put all this together, and one can, following this recipe for nonlinear, from linear out of equilibrium thermodynamics, one can calculate the entropy production, and as expected, it takes this form here, so it takes the form of the sum of coupled forces and fluxes. Okay, and I can look at their conjugate pairs. and there is the surface shear, so is conjugated to the tension tensor on the surface. This kind of coupling is equivalent, the two-dimensional equivalent of the coupling for a three-dimensional fluid between the stress tensor and the gradient of flow. But I also have other pairs. The bending moment tensor is coupled to the rate of change of curvature tensor. Okay. The molecular field is coupled to the polarity dynamics. Those are two objects from liquid crystal physics, which are already known there. And the surface is active, and so the rate of ATP consumption is coupled to the chemical potential of ATP hydrolysis. Okay, and this is a term that brings the system out of equilibrium because it sort of describes the fact that there is chemical energy available to the system. Okay, and then I follow this recipe and expand the fluxes into forces. And so, for instance, I can expand the tension, the bending moment tensor, and the polarity dynamics into their contribution. And here I find again this epsilon here, which are non-zero only if the surfaces have the appropriate broken symmetry. For instance, this term can only exist in a surface that has up-down broken symmetry. Okay, and then I write all the possible terms that couples the fluxes to forces, and you can see that there are many of them. And so, of course, we're not going to write all of them at the same time. That would be too much work. But nevertheless, okay, it tells us what's possible. So on the good side is that there are many physical effects that can be captured by these equations. Okay, but then we need to choose which terms are appropriate for different physical situations. Okay, which is what I'm going to do now. Time.
So there is a question online.
Yes?
You want to answer it now?
Yes.
Okay, so Camilo Andino asked, thanks for the nice talk so far. I was having a small doubt. Is the amount of pseudoscalars you presented related always with the dimension of the rotation group of the system?
To the dimension of the rotation group of the system?
Is the amount of pseudoscalars you presented related always with the dimension of the rotation group of the system?
So, I don't know if this is referring to rotation group in 2 dimensions, but to define the pseudoscalar, they exist for a surface which is embedded in 3 dimensions. They will not exist in a pure 2-dimensional surface. So one needs to use the fact that the surface has a finite thickness. I don't know, I mean, I haven't looked at surfaces of dimension n, so I don't know how this generalizes to. Okay, so let me start with a simple application, which is cell migration powered by cortical flow, which is some work we have done some years ago, but it uses a version of this theory, which is sort of the simplest choice you can make, where out of the many terms I was showing before, one just keeps 2 terms. One that describes which looks like the Newton equation, the equation for a Newtonian fluid, which introduces a viscosity. And this now is an active stress, so it depends on the ATP chemical potential. And it's just, in a 2-dimensional surface, it's just proportional to the metric, which means that it's isotropic, it's the same in all directions. Okay, and this equation can be used to describe the flows of the cortical actin on the surface of the cell. And why? Because one expects this network to be fluid, because it's a polymer network. If one stretches the polymer network, there are cross-linkers which are going to detach and let the material flow. So there is in principle no elastic stresses that remains because of turnover of the replacement of the material. And also because there are these myosin molecular motors which are working out of equilibrium using the energy of ATP hydrolysis to generate stresses. Okay, just a simple picture is, let's imagine that we have a piece of this cortex of this actomyosin network, and we have more myosin molecular motor in some region. According to this equation, they are going to generate a higher active tension, and therefore there's going to be a flow for these 2 terms to balance. And this flow, flow is in principle going to be predicted by this constitutive equation. So this is a practical example here, looking at a migrating cell in a channel. So, okay, so this cell is moving in the channel. Okay, it's confined. And if we look at a cross-section of the actin cytoskeleton, we can see that it's mostly on the cell surface, which is what I was saying, that okay, most of the actomyosin cytoskeleton is close to the cell membrane. And I hope this one will work. No, okay. Okay, and so if one looks at the distribution of myosin, one sees two things. One, which is that there is more myosin at the back of the cell than at the front. And also, if one could see the movie, we would see that these myosin spots, they are not moving relative to the extracellular channel, so they are moving inside the cell from the front to the back of the cell. So there is a retrograde flow of the cortical actin from the front to the back. Okay, and one can then make a model of this using this active surface model, where now the cell is represented by a two-dimensional surface of active fluid, which has a viscosity, and there is also this active tension, which is not the same everywhere, so it's higher in the front than in the back. And then this is going to give rise to a flow because of the higher contractility in the back. And since the cell is confined in a channel, there is in general going to be a friction force that acts against the flow of the cell surface. Okay, and also the cell needs to push the fluid in front of it in order to be able to move, and that creates an external drag that opposes the cell motion. Okay, and then this is a relatively simple model that one can solve. And okay, and if one solves the model, one sees, as expected, that there is a flow from the front to the back, which is resisted by a friction which pushes the cells forward. Okay, and then the theory allows to predict how the cell is going to behave as a function of the friction coefficient. One can calculate the cell velocity here in blue and the magnitude of the retrograde flow. And there are different regimes, okay, which we can observe experimentally. At high friction, the cell is moving, but this is the retrograde flow measured with respect to the lab reference frame, and so there's almost no retrograde flow. At very low friction, the cell is not moving because there's not enough friction, so it cannot engage with the material confining it. Instead, it stays fixed, and the actin is going backward from the front to the back, but does not result in any motion. And in between, there is a regime where there is a retrograde flow and there is motion. Okay, so, but now coming back to epithelial mechanics, okay, we need to move away from this simple version and have select terms which are going to be more appropriate to describe epithelial tissues. Okay, and this is what I'm proposing as a choice that's going to be appropriate for epithelia. One can recognize the same term as we had before, the viscous tension and the active tension. But there are additional terms, notably now we can take into account the internal bending moments because the epithelia are thick. So they are sufficiently, so therefore approximating them by simple surfaces is probably going to be incorrect. Because they are along the cross-section, they are structured. So, okay, and then why making these choices? One aspect is that, in principle, if one pulls on a tissue, we expect this to be fluid because there's going to be topological rearrangements, as we were discussing before, which corresponds to the disappearance of some junction and reappearance of some junctions. And these topological rearrangements, by which cells change their neighbors, they allow to relax the elastic stresses in the tissue and they allow it to flow. However, this second term here describes the resistance to change in area, and we expect it to describe the fact that the tissue is elastic with respect to changes of area, because if there is no cell division and cell death, if one pulls on the tissue in all directions, so isotropically, the cell area increases, and if the cells have a preferred area, they will then exert a restoring force. Okay, and then if we look at the terms that are included into this bending moment, there is a passive term with a bending modulus, which is very well known in the physics of lipid membrane, which says that the epithelium is going to resist curvature changes. And finally, there's this active term, this active bending moment, which is not really like an active tension, it's really an active bending moment, so it's a force that's trying to actively turn the cross-section of the tissue. And this can be generated if, for instance, you have an apical stress, you have a basal stress, which are different, and then this is going to create an active torque, which is related to the difference of this active tension apically and basally. Okay, and with this in mind, we can return to the question of the tumor growth, and we can now do a continuum calculation where we take a cylinder and we say now this red piece is a tumor, so it wants to grow, so it has a new preferred area. But because of the redistribution of myosin apically and basally, there is also a different active torque or active bending moment in these two regions. And then one can do an analytical calculation and look at the resulting shapes. And depending on the radius, we see again this sort of generic result that there is a threshold of radius below which the deformation is positive and above which the deformation is negative. One can find analytically the threshold, but maybe importantly, one can also see where the effect comes from, why there is this competition. Okay, and the reason is that there are two effects. One is there is cell proliferation, and this leads to a local enlargement of the tube because one is simply adding more cells. The other effect is a tendency of the system to spontaneously curve, so to move towards the inside of the tube, and that's due to the difference of active bending moment, or to say it differently, to the redistribution of apical and basal surface tension. And when the tube is sufficiently small, this effect dominates, because when the cells divide, they're always adding the same number of new cells, and the overall effect is much bigger in a small tube. Also, it's more difficult to deform a small tube than a large tube because of the bending rigidity. And these two effects combine and create the fact that in an infinitely large tube, the tumor always wants to go inside, but in a small tube, the proliferation is dominating and the tumor grows to the outside. Okay, so so now we have this theory for epithelia. Here I've applied it to tumors, but we also wanted to look more generally what we can predict for this theory. And okay, our general motivation is to look at morphogenesis and also organoids. So these images are showing a few examples of organoids, which are really remarkable structures. So these organoids, they come from stem cells, which are grown in vitro. And depending on the cocktail to which they are exposed, they spontaneously form this remarkably pattern and deformed structures. Okay, and depending on and they share some feature with the adult organ, and depending on the cocktail, one can bias them towards different types: retina, intestine, etc. Okay, and it's kind of a nice question to understand how this self-organization comes from, how without any external information, the system manages to generate this. Okay, and here I'm asking a much more modest question, which is that usually these organoids grow first by forming a cyst, so isotropic shell, so an epithelium which is empty at the center, and which is spherical, so it's the structure with maximal symmetry. And then there is a symmetry breaking event, which, as described in this paper, is usually due to signaling. So there are exchange of molecules, which allows the system to choose an axis. And then one can ask the question, once this axis is chosen, the tissue is going to deform, but we then expect that depending on the cell fate decision taken through signaling, how we can ask how the resulting mechanical patterning is going to result in the deformation of the tissue. Okay, and here I'm simplifying this by saying I take a sphere, I have two tissues with different types, and I can ask what sort of deformation I can expect. And if I just assume that there are differences of active tension, which is the simplest thing one can expect, that a tissue is more contractile than another, then there's not going to be any 3D deformation. The theory tells us that one tissue is going to constrict and the other one expand without an overall deformation. But now if I have a patterning in the active torque, in these active bending moments, maybe I can expect three-dimensional deformation. Okay, and so this is what we have done. So this is an example of a simulation using the equations for tissues that I was showing before, where here one has taken the red and the blue region and assumed that they are under different active torques. Okay, and this is the color code is for the velocity field, and one can also see the arrows indicating the velocity. So as a result of the difference in active torque, there is a budding event. So one of this passive part here is sort of evaginating out of the tissue. And this is generally what happens. This is a phase diagram that shows on the x-axis the magnitude of the active bending moment in the active region, and on the y-axis the size of the active region. And most of the time there is either invagination or evagination of a bud. This is in fact something that is seen in intestinal organoids. You can see a set of images here. This is from the Liberali lab. And you can see that these cysts are growing, and at some point they are indeed forming a bud. This has been described also in this paper theoretically using a similar approach, saying that different parts of the tissue have a different spontaneous curvature, which is what these active bending moments are generating. Okay, but looking at the phase diagram, one can also look what happens after this line. And after this line, there is a transition, there is a shape instability. If one crosses this line to that direction, then there is a sort of strange shape that emerges, where the surface rubs onto itself and self-intersects. But with the model, we cannot predict what's going to happen after self-intersection. And if we look at the open dotted line here, instead we have a formation of a neck. So if one goes past the threshold, the neck is going to constrict in finite time. So presumably this would result in a separation of the surface into two pieces. And right on the line, there is a funny solution, which is an ideal neck. So this is a neck that connects two spheres, but the neck has a size that goes to 0 exactly on the line. So it's more of a theoretical interest because, of course, the epithelium has a finite thickness, so there's going to be a self-intersection before this line. Okay, but so looking at this diagram, I mean, clearly the sets of shape we obtain is quite limited. So we thought we would also investigate what would happen with nematic active epithelial shells. So assuming now that because of cell polarity or cell nematic order, that there is a nematic field on the surface, and this nematic field is assumed to minimize an effective energy, which has two parts. The Landau-Deugen part is saying that the system is a nematic phase and wants to be ordered, and the Frank free energy is the same distortion energy that I was writing here. It's saying that the director wants to be parallel to each other spatially. Okay, and the ratio of these coefficients determine, so we look at axisymmetric spheres, so then there must be defects at the top and at the bottom. And there is a defect size in which, so there must be two topological defects. And at the center of the defect, the order must dissolve, and this dissolution of order occurs on a characteristic length scale, which depends on these parameters. Okay, and then now we can explore the effects that can be introduced by this nematic field. For instance, the fact that the tension tensor depends on the active nematic tension. Okay, and if we do this, we see that already the isotropic shapes can deform. So if the orientation of the order is meridional, like this, meridional, yeah, not sure. Then the shape progressively flattens and forms a disk, which looks like a bilayered disk. And this can be understood, I think, because if you look at the surface from the top, there is this radial order that emerges, and the internal tension of the system is creating a tension along these lines. And to me, it makes sense intuitively that this would tend to flatten the spheres. Okay. then if the orientation of the nematic order is the other way around, instead the surface elongates, and at this point there is a bifurcation, so the surface loses stability. And above the bifurcation, what happens is essentially what looks like a cell division, so there is an instability and there is a separation at the equator of the shape into two parts. But this is not exactly like a cell division, because in the cell division, myosin and actin are recruited at the equator and are pitching specifically there, whereas here the nematic order is uniform, but despite this, there is this physical instability that divides the system. And if the activity is very high, the central constriction creates a long tube, but this tube is not stable, and eventually there are necks that forms and splits it into several pieces. Okay, so that's the nematic tension, and the last thing we tried was to now assume that there is a nematic torque which is created in the system, which is oriented by the nematic field. This could arise, for instance, if there would be a nematic order on two parts, on the apical and basal part of the tissue, with different contractility. Or it could be generated if we had two layers with perpendicular order, okay, which are both contracting but in opposite direction. So this looks like the pseudo-nematic phase I was showing earlier. And in fact, so you have heard about this yesterday, but there is this very nice paper from a Kinneret Keren, which shows that in Hydra, the organism is made of two tissues. And if one looks at actin fibers in these two tissues, they are actually arranged orthogonal to each other. So this is the kind of situation that could create this nematic torque. And if the other interesting thing they find is that there are defects in the orientation of the actin fibers, which are specific points on the body plan of the organism, so at the tip of the mouth or at the tip of the cylindrical tentacles. Okay, and now when we take this into account, this is actually what we find. So now the active region here is under high nematically oriented active bending moment, and at the tip of this tubule that spontaneously formed, there is the +1 defect. So it's kind of the same structure that emerges. And one can understand the size of this tube by looking at the expression for the torque, which has a passive term, which depends on the bending modulus, and an active term, which is oriented by the nematic field. And if one looks at this balance, one can see that the radius of the tube decreases when the magnitude of active effects is increased. Okay, and one can also look at the phase diagram. So again, this is the size of the active region. This is the strength of the active nematic torque. And you can see that one generates these tubes of different lengths, but also on the other side, the tubes are growing now not to the outside, but to the inside of the spheres. And finally, they can reach this red line, and that's the situation where the tube enters the sphere and collides on the other side. And this is interesting because some primitive organisms, like sea urchin, undergo a gastrulation phase that looks like this. So they are spherical, but they have invaginated tubes that goes to the other side and then fuses. And then this overall sequence of events transforms the sphere into a torus. And this fundamental topological change is essential to the development of organisms. So here we have a possible mechanism that can generate this topological deformation. Okay, so overall, here we have sort of looked at, with relatively simple hypothesis, as the zoology of shapes that we could obtain. So we see no shape change with isotropic active tension. We see budding. We see we can generate a flat disk or a division, and we can generate these tubes and transform a sphere into a cylinder. And I guess the question more generally is whether biological systems are using this space of shapes to operate. Okay, I wanted also to say a word about chiral effects. So one additional way that I can change these equations that I was showing is to take a tissue which is now flat, but is also planar chiral, meaning that I can draw a clockwise or a counterclockwise direction on it. And this can, this is a bit of a special situation, but if you have a tissue which is on a substrate and the difference between apical and basal axis and is also chiral, then it has both up-down broken symmetry and a broken chiral symmetry. And as a result of the combination of these two, the planar chiral symmetry is also broken. So in fact, they are all broken. But that means that the planar chiral effect can emerge. Okay, and then I take again my equation, but I add this extra term here. So I recognize the same structure. There is a viscous part, there is a nematic active part, and there is a planar chiral part. Okay, then I can say, okay, if I have no planar chiral effect, I have a simple situation where I can have a nematic axis in the system, and there's going to be a tension oriented by the nematic axis. If I add this active planar chiral effect, now I can define what clockwise is on the tissue, and so I can have an axis of contraction of the system which is not oriented with the nematic axis, which has some tilt relative to it. Okay, so I have, for instance, the cells are elongating in a direction, but instead of contracting along the direction of elongation, the tissue can constrict, but with an angle. That's general argument based on symmetries. Okay, and I can also write an equation which I have not talked about too much, but which is going to tell me what the axis of elongation of the system is. And there are several terms. This one minimizes the spatial distortion, the distortion energy. There is also a general term that couples the flow and the director, which says that if I have a flow in the system, the nematic orientation can change. This is a classic term in a liquid crystal. And there can also be a planar chiral active polarity rotation, which only exists for planar chiral active system, because once I have an axis of rotation, I can, if I know clockwise on the tissue, I can imagine that active effects are going to try to rotate the polarity. Okay, and so now I can compare this with, again, the work from Pascal, which shows, which here is looking at the stripe of tissue. And so the tissue is cultured in a micropattern glass substrate, so that is confined with a given width. And here, when cultures elongated cells, and the cells tend to align parallel to the boundary of the stripe, and then one may expect that they will nicely stay parallel to it, but above a threshold length of the system, there is a transition, and the cells start to tilt. So at the center, they are not aligned with the stripe. They make some angle, and there is a spontaneous flow. Now this can be understood by a model which was first proposed by Raphaël Voituriez and co-workers, which is analyzing the behavior of a tissue confined, or an active fluid confined in a stripe. Okay, and there is an order of the system, and if the system is sufficiently active, the order, instead of staying parallel to each other, which will minimize distortion, undergoes an instability, starts to tilt. The tilt creates an active tension, which creates a flow, and the flow feedbacks on the tilt by a flow coupling between director and flow, and so this instability leads to an overall deviation of the tilt. Okay, and then one can obtain a curve like this, where there is a length of, as a function of the length of the stripe, the angle at the center of the stripe is initially vertical, but then flips in two opposite directions, which are equally likely. So this is what this movie are showing. It can flip to the left or to the right. But if we introduce planar chiral effect, this pitchfork bifurcation here becomes imperfect, and there is only one branch that is chosen that one can follow continuously. This is a consequence of the fact that clockwise can be favored, for instance, over counterclockwise. So these two possible orientations are not equivalent anymore. And this is something that is seen in the experiment. this is now the angle at the center of the stripe as a function of the length of the stripe. And you can see that it only goes down. So there is a signature of this planar chiral broken symmetry in the fact that the angle chosen by the tissue is always in one direction. Okay, and there's one more theoretical prediction which maybe is interesting, which is that if there is no planar chiral effect, okay, we have this pitchfork bifurcation. If we have a weak planar chiral bifurcation, then the weak planar chiral effect, there is an imperfect bifurcation. But eventually, this curve will saturate. But then there is another transition for a strongly planar chiral system. Instead, the angle at the center gets bigger and bigger. And what actually happens is that above this transition, yes, okay. Above this transition, the system transitions to a situation where the order rotates. And so what happens here is that the order is forced to be along the wall, at the boundaries of the stripe. But at the center, it's rotating. But as it rotates, it accumulates rotation in between the wall and the center. And so there are loops of the order which accumulate. And it's like twisting a coil. The coil spring is like twisting a coil spring. At some point, there is too much distortion energy, and the system stops its rotation. Okay, I don't know if this has been observed in tissue, but I wanted to mention this phase because it exists. Okay, actually, I will stop there.
Thank you, Guillaume.
Thank you.
So I start in the last part. I'm sorry. The spheroid at 2 + 1 sides, right?
Yes.
Why did you? I mean, you choose that initially, or can you have 4 + 1/2, and what would be the consequence then?
Okay. Yes, a good question. So we chose it to be axisymmetric, so that forces the system to have 2 + 1 defect. If it was really a pure nematic, it should be 4 + 1/2. So this would actually work if there was an additional polar field which makes the defect, or we're actually describing a polar field which then make the 2 + 1 defect the minimum energy state. Also for the non-axisymmetric solution. Yeah.
That's something you prescribed initially.
Yes, that's prescribed initially. Yes. Yeah, it would be nice to also look at the general shape, not imposing axisymmetry, but it's much more, I mean, to explore the phase diagram would be more complicated.
So you showed the hydra that we saw yesterday. So everything will be very familiar with nematics after this set of lecture. Is it the same process by which muscles assemble around the intestine, because they show the same crisscross geometry? And is there a particular reason why it happened only on tubes and not, as far as I know, and not on other geometries?
Okay.
Two nematic layers that sort of crisscross.
Okay. So I didn't know this existed around the intestine.
Right, it's around the gut and around the arteries, by the way. Too.
Okay.
Yeah, it's very common. I don't know, but it's very spread in many, but it's always in this geometry.
Okay, so.
The tubular geometry.
Okay, so actually that's very interesting because if we take a sphere and we implement this structure of this double orientation, it makes a cylinder. So maybe there is a link with this is the right structure in general to make a cylinder. Yeah.
Okay.
So there is a question from a remote participant, this Rolling Romain. So thanks for this lecture. I was wondering, in all the deformation you showed, body and so on, you were imposing a constant value?
A constant value, yes.
Volume?
Volume. Okay, we have two. Either we impose a constant volume or no difference of pressure. And there are slight differences. For instance, the tubes, they only appear with no difference of pressure. if we impose, when we fix the volume, the deformations are a bit more complicated, as one might expect.
Thank you.
Thanks.
If we go back to this example of Hydra from Karen, if I remember correctly, in fact, the width of these fibers are different on the inner and outer layers. So I wonder whether this is something that you take into account, or that somehow, I mean, the fact that one is inside and the other one outside, you can be predicted through this sort of framework that you propose. And what can you tell us about this?
Okay, so I think, so if it was a pure, okay, it's a good question. So I think, so if there are some differences between the two layers, then it's not a pure pseudo-nematic phase. There is also an actual nematic order. And in this study, we have separated the effect onto tension and bending moments. And intuitively, I think if the two layers are not equivalent, that's also going to generate a tension. And putting them together is something we haven't tried. So we should try it. So I don't know if that answers your question. Or...
I was wondering, whether on the basis of the sort of symmetrical argument of symmetries that you propose, you could, for instance, predict, that thick bundles would be inside or outside. I mean, this sort of.
Yes. Okay.
How can you?
Okay. So
link the breaking of symmetry in that case with some of these symmetries argument that you proposed.
Okay. So the inside-outside orientation, there is one particular choice if the fibers are contractile, which makes a tube. If the fibers are like this, the tubes goes to the outside. If they are like this, it goes to the inside. So if the fibers are contractile. So that's one condition imposed by symmetry. Now, whether the filaments are shorter to the inside or outside, I don't have any clear intuition of what this would do. Yeah, okay, it sounds like a good question. Yes, so I wanted to thank them as well as my collaborators. Yeah.
Do we have any questions?
Thank you, Guillaume. So experimentally, for the first part, when you talked about this pancreatic cancer and the bulges and so on, so you talk about a critical radius that you can have the bulge inside or outside. So in reality, is there a region where you can have both? I mean, is there a radius?
A coexistence region? Yeah, so when we quantify, of course, there is some noise, and if you plot the deformation as a function of the radius, there is a distribution, and there is a radius at which, on average, it's 0, and so it can be positive or negative. Yes, that's.
So in these regions, locally you have different tensions?
Yeah, so we didn't check. Yeah, or I didn't at least. Yeah, it's a good point. Yeah. On average, it's true, but whether it's really in the same region, this I don't know. Yeah.
Thanks.
Yes.